Hello everyone. Good morning. So let's start to discuss Fermenter. So this particular topic is useful for industrial microbiology, industrial biotechnology, microbial biotechnology, bioprocess engineering, and many more related to the biological processes. So first, it's very confusing. There are two words, fermenter and the fermenter. Actually, these are interchangeable. Both both may be used for a vessel or a organism. Usually, what I recommend to my student is when it's a OR fermenter, then it refers to the organism which is causing the fermentation, which is responsible to carry out the fermentation process. And the fermenter ER is referring to the vessel in which a particular process, or you can say the fermentation process, is going on. So, a fermenter is basically a device in which substrate of low value is utilized by living cells or enzymes to generate a product of high value. It's a containment system. It's designated to right environment for optimal growth and metabolic activity of the organism. Now, we will talk and we will go deep into this. This can be open as well as it can be a closed one. But before going to this, let's recall the basic functions of the fermenter, why the fermenter is there, and what is needed for. Very first thing is it provides a controlled environment, and this controlled environment is optimum for the product yield. And your product can be your enzyme, can be your secondary metabolite, can be biomass itself. And as we know, the fermenter is most widely used in the industries, and it's a main component of bioprocess engineering. So we must think on the economical concept of this. So the power consumption must be minimum in case of the fermenter. There must be a facility for sampling so that if at any time contamination occurs, we can stop the process, we can save our further media, we can save our electricity, we can save our manpower. Evaporation loss is one of the main things when we are considering any bioprocess. So it must be designed in such a way that it is as low as possible. And about the internal surfaces at the joints, it must be as smooth as it can. Because if it's not smooth, the bacteria, which are common contaminant of the bioprocess, forming the biofilm, they may harbor there, they may grow there, they may form the biofilm. This is thereafter, it's very difficult to remove from the process and it will continuously contaminating the one batch and another batch. Yes, it must provide an adequate mixing and the aeration for optimal growth. So this is a generalized, if you can see, it's an ideal, or you can say what are the different accessories which is related to the fermenter. So you can see here, this is a overall fermenter. There's a vessel. There is a motor which is fitted with a shaft, and it is responsible for rotating the impeller, or you can say agitator. Then we have the compressed air coming from the compressor, and it is added into the vessel via the sparger. Then we are having the jacket outside this. This is a jacket in which cold or you can say steam is coming out, and it is going to. This is used for the temperature controller. Then we can use the steam to sterilize the harvesting pipe or to autoclave this. So here the different ports are there. These are the ports. One is antifoam, another is for acid and base, then another for the filtered waste gases. Then for the nutrient or the inoculum, we are having the sample. Then this is the media inside this, and then this is a dissolved oxygen probe. There are three probes, four or five ports, three probes. This is used for the oxygen, dissolved oxygen concentration, so that we can regulate the supply of the compressed air. Then this is a pH probe, which will redirect whether the acid or base is added if pH is going down or up. And this is a temperature probe, which is regulating the cold water inlet so that the optimum temperature can be regulated. Then the harvester pipe is there. Okay, so this is a generalized one, generalized fermenter. It must be there. So there must be the ports, there must be accessories including your impeller, compressor. One more thing which is lacking in this diagram is your baffles, which may be added to here, which are serving as one tenth of the total diameter of the fermenter or the vessel. And the working volume of an ideal fermenter is around 70%, or in another word, two-thirds. But exactly the two-third is around 66.6%. So usually, you can say when we are handling any bioprocess, it's usually 70%, and 30% of the area is kept free from the medium so that it will not touch any of the gasket or the port, which are usually the source of the contamination. So we are stopping here because I will come back with short 1 or 2 minute videos. So stay there, stay in touch, please subscribe and follow. Thank you. Thank you.